Hello dear learners, in this video we are going to talk about the various kinds of mutations which can be observed on the chromosomes. Now there are two different types of mutations which can be observed on the chromosomes if we categorize into broader base. One of them is if any kind of structural mutation is observed in the chromosome, means the chromosome is present but its structure is altered. The second kind of mutation is related to the numbers of chromosomes, that whether the numbers of chromosomes are increasing or decreasing. So there are two kinds of mutations which can be seen in the chromosomes. So if I talk about the structural, so structural ones are the those where the structure gets altered, and then the number ones. And in numbers also we have two types. One of them is aneuploid condition and another one is polyploid condition. So in the aneuploid condition, what happens is generally one of the chromosome, only one or two or three chromosomes, they get added or subtracted. But in polyploid, the whole set of, like suppose if we have 23 chromosomes in the haploid set, so whole 23 and again 23, this much amount of chromosomes will be coming. So that is the condition of polyploidy. Whereas in aneuploidy, only one of the chromosome gets duplicated, but we will study it later on. First of all, let us see that what can be the structural variations which can be observed in the chromosomes and which can lead to the mutations. So moving on, yes, there are four major types of structural rearrangements on the chromosomes which can occur, one of them being the duplication. Now as the name suggests, suppose we are taking this EF gene, so this EF gene gets duplicated, so that time it is the kind of duplication of the chromosome. In the deletion part, this EF gene will be removed from the chromosome. In the inversion part, suppose we are taking these three genes present on the chromosome, let's say D, E and F. So the D, E and F genes, they get inverted. So there is a 180 degree inversion means exact the mirror image which has been formed. So in this mirror image plus the 180 degree, so one thing that you have to write is that D, E and F should be in the inverted form, just as to show that it has totally rotated 180 degrees. And then lastly we have the translocation, when we have two different chromosomes and one or the segment of genes, they get inserted into another chromosome, and the another chromosome's genes get inserted into the first chromosome. That is basically we have the chromosome translocation which is occurring. So these are the four types of mutations which can lead to the change in chromosome structure. So the type 1 mutation, that is chromosomal structural mutations, consist of these four subtypes. So now we have to talk of all of them in detail. So firstly we'll talk about the duplications. Now duplications are those wherein a part of gene in the chromosome, it gets doubled. Now these duplications can be of three types. One of them being the tandem duplication, one of them being the displaced duplication, and one of them being the reverse duplication. In the tandem duplication, the duplicated segment of the chromosome will lie just next to the original segment. Suppose if we say that there is the original segment whose duplication has to occur, suppose EF duplication has to occur, so the duplicated EF will just lie next to the original or the parent copy of that gene. So this is the tandem duplication. The second can be the displaced duplication. In the displaced duplication, the duplicated segment lies far away, means whatever it has been duplicated, supposedly the EF has been duplicated, so new EF gene is located far from its parental genes. And in the reverse duplication, what happens is that the EF can either lie far or either can lie close, but it will be inverted in the inverted or the reverse position. So we call it as the reverse duplication. So there are three broader categories of duplication, that is tandem, displaced, and reverse duplication or the invert duplication. Also we have two terms here, one of them being the homozygous duplication and one of them being the heterozygous duplication. In the homozygous duplication, the two chromosomes which are present in a cell, both of them contain that duplication. So the chromosomes are said to be homologous. Suppose this is my one chromosome and this is my another chromosome. Now suppose this A gene is getting duplicated. So this A gene will be getting duplicated in both the chromosomes. So that time this is your homozygous chromosome. In the heterozygous chromosome, one of the chromosome will remain normal and one of the chromosome will have the duplication, like in this case. Now suppose we have a pair of heterozygous chromosomes, like here we have one of them being the normal one and one of them being the duplicated one. Let's say that we have to bring them into meiosis so that the exchange of genetic material can take place and finally the gametes can be formed. So if we have to form the gametes from these two chromosomes, so how can we do that? We have to first of all align them in the prophase 1 phase. Only after that we can form the gametes. So it is just a very normal biology. Just try to understand that there are two chromosomes present in the cell and now we have to do the meiosis. For doing the meiosis, we have to first of all align the two chromosomes so that the genetic material can be exchanged in the pectine stage. So for the exchange of this genetic material, the two chromosomes have to align with very close to each other. For this alignment phase, we have the prophase 1. So when in the prophase 1 the two chromosomes have to align, now here is a difference. One of the chromosome is a normal chromosome while the other one has the duplication. So how to align both of them? The alignment can only be done when all the genes are properly oriented in both the chromosomes. So this can only be done when a loop is formed of the extra duplicated chromosome. Because for this E and F, we do not have any matching pair in the another chromosome. So this E and F chromosome has to be looped out. So this will happen during the alignment in the prophase 1 stage. So now this is your cellular marker. Whenever in the cell during the prophase 1 stage, if you see that there is a loop on the chromosome, that means that chromosome consists of certain duplications. So this can occur in the duplication also. Now there is a very good example here which is there of the Drosophila. The Drosophila have a bar mutation and it is a kind of duplication. Now in the normal Drosophila we have the chromosome X. This chromosome X consists of a gene which is called as a bar gene or the bar region. Ideally, one chromosome X has to only contain one bar gene. And for the females there is XX, so 2X should contain one bar region each. Now what happens is sometimes this bar region can get duplicated. If the bar region is getting duplicated only on one of the X chromosomes, then the eye from the oval shape will become short or will become thin. If there is a bar mutation on both the chromosomes, then it will become a bar shaped and small. And finally, if there is a double bar mutation on any of the chromosome, means the bar gene is repeating 2 times again, that time the eye is very minimal eye. So always remember, whenever the bar region gets duplicated, that time the eye size decreases. So there are the 4 conditions which can be possible. How can this bar gene be duplicated? Suppose we have the wild type chromosomes, that is the X chromosomes, and we have to do the crossing over of these X chromosomes. Now suppose the two X chromosomes are not properly aligned. In that condition, this region will cross over with this region and here this bar can move this way. So that time during the crossing over, one chromosome will contain 2 bars. So this now has the bar mutation. And the other chromosome does not have anything. So it is again a mutation. It is a kind of deletion mutation. So this is also a problem. This is also a problem. Now suppose this bar mutated chromosome, we take two such bar mutated chromosome and again same, we have this that they do not have the proper alignment. Now the crossing over will be in such a way that one chromosome will get 3 bar regions, that is it will be doubly mutated, or we say it is double bar mutation. We can represent it by X dash dash, and one of the chromosome will only contain normal X because normal X has to have this bar gene, so it will become normal. This will become again the wild type, but this will contain a double bar mutation. And whenever there is a case of double bar mutation, you can see that which kind of eyes are produced. So generally what happens is wild type fruit flies have the oval eyes. Then the flies which are heterozygous, they contain small and bar shaped eyes. Similar is for this one. who have the homozygous for the bar mutation, here also they will have the bar shaped eyes, and when this mutation gets doubled and again doubled, that time their eyes become very small bar shaped eyes. So this can happen. So we are only considering one example here. Then there is a term which talks about the segmental duplication. Now segmental duplication is common in humans. What happens is there are about large duplications of certain regions which are approximately of 1000 base pairs, and these get duplicated again and again. So this is generally normal in the human chromosomes, and this is only the one which is the important source of evolutionary novelty. Now this can be intra as well as interchromosomal duplication, which is observed in the humans. So segmental duplication is nothing but the genome which consists of the numerous duplicated sequences. Coming on to the next type of structural mutation subtype, that is deletions. So now deletion is opposite of duplication. What happens is generally one or the other segment of the gene gets deleted. Now if you try to compare the harmful effects of duplication and deletion, you will come to this conclusion that deletions are generally more harmful than the duplications, because in the duplications only one of the particular gene is getting duplicated. So that is the protein which is produced now will be more in amount, and this protein which is more, or the dosage of this protein which is more, may be a problem, but not as severe as in the deletions, because in the deletions the whole of the gene is removed, and when whole of the gene is removed, that protein cannot be formed, and this will cause many of the deficiency disorders. So this is your chromosomal deletions. Now if we talk about the deletions, there can be two different types of deletions, one of them being the terminal deletions and one of them being the uh, interstitial or the intercalary deletions. Now see what happens generally in the terminal and the interstitial or intercalary deletions. Yes, if you see here, F and G genes, they are removed, and these F and G genes were present from at the terminal position. So if they were present on the terminal position and they have got removed, so this is what we call as terminal deletion. Similarly, if there is a gene which has been removed from in between, suppose there was A, B, C and D, and now the another has only A and D. So that time this B and C from in between have been removed. So this is called as intercalary deletion. So there can be two types of deletions. Now again, as we were talking about the duplication, during the prophase 1 there has to be this phenomenon that if the F and G genes are not present, then we have to make the loop out of it, because so that it can be easily pairing with this part. Now see, in the deleted part the F and G genes are not present. So if the F and G, okay, wait, G is present but E and F are not present, so E and F have to be looped out. So suppose this is my chromosome 1, it consists of here C, this is a centromere, so C, D, E and F, and then here we have G, and this is your chromosome 2 which consists of C, D and G. Now this can pair, this can pair and this can pair, but EF cannot pair, so it has to be looped out. So again this can occur in the deletions as well. Now we have to talk about the concept of haploinsufficiency. It is a condition where only one functional copy of the gene, it becomes insufficient to produce the required dosage of a product for the development. Suppose we talk about a certain gene. Let's talk about uh, a gene named A. So we know that the humans are diploid and it will contain two copies of the chromosome. Now suppose both of the copies contain this A gene, and both of the A genes are coding for a protein. Now if one of the A gene is absent, then the protein would not be coded and the protein dosage will become half. So this time the effect which has to be produced by this protein cannot be produced because its concentration is very low. That time we say that it is a haploinsufficient gene. Another concept comes of the pseudodominance. Pseudodominance occurs in the heterozygotes. It is a condition where the recessive allele is expressed because the dominant gene gets deleted. Suppose I take the example here only. Let me remove this. Okay. So let's take two copies of a chromosome. Let me increase its width. Yes. Suppose I have this chromosome 1 and this as the chromosome 1's another copy in the diploid cell. Now suppose this had a gene which is A, capital A, it has capital A allele of this gene and it has the small a allele of this gene. Now due to some deletion this capital A got removed. Now only small a is there, so it will be expressing itself. In that condition this capital A has been removed and only the recessive now expresses. This is called as pseudodominance. So this is another phenomenon which is associated with the deletion thing. Now there is one very important disease in humans which is the Cry-du-Chat syndrome. Now Cry-du-Chat syndrome is here. What basically happens is in the chromosome 5 there is a 5p arm. This 5p arm is present on chromosome number 5. Now this 5p arm gets deleted, and when this particular deletion happens of the short arm on the chromosome 5, this results to the structural mutation in the chromosome, and the syndrome is referred to as the Cry-du-Chat syndrome. In the Cry-du-Chat syndrome, there is a peculiar cat-like cry of the infants which have this syndrome. Along with that, they also have the microencephaly, which is the small-headed condition, and they have certain intellectual disabilities also. Another example is the deletion of a part of short arm of chromosome 4. So if there is a part which has been deleted on the chromosome 4, it results into a disorder which is referred to as Wolff-Hirschhorn syndrome. It is characterized by the seizures and severe mental and growth retardation. Another deletion, if occurs on chromosome number 7, it causes haploinsufficiency because the required amount of protein is not produced, and that also constitutes some of the disorders. And this particular syndrome is known as Williams-Baraitser or Williams-Baraitser syndrome. So we have talked about the three important syndromes, one of them being the Cry-du-Chat syndrome, one of them being the Wolff-Hirschhorn syndrome, and the third one being the Williams-Baraitser syndrome. The Cry-du-Chat syndrome on chromosome 5, Wolff-Hirschhorn syndrome on chromosome 4, and Williams-Baraitser syndrome on chromosome 7. All of them are the cases of deletions only. The third type of phenomena which we can observe in these structural rearrangements being the inversions. Now in the inversions, what happens is generally the segment of gene or the segment of chromosome which consists of certain genes, they invert themselves and turn into 180 degree. Now when I talk about 180 degree, as I told you earlier also, suppose here I have 5 genes A, B, C, D and E, and the B, C and D gets inverted. So here we will have this way. There is A, then B, C and D will be like this. B, C and D, total inversion, and then we will be having the E. So here we have the 180 degree inversion. Now these inversions can be of two types, one of them being the paracentric and one of them being the pericentric. In the paracentric inversion, the centromere is not involved, just as this what I have drawn here. Here also the centromere is not involved, so this is the case of paracentric inversion. In the pericentric inversion, the centromere is also involved. Now suppose if I talk about a particular chromosome which has these genes, here I have A gene, B gene, C, D, E. Now B, C and D are getting altered. So it will now become like this. Also let's say this A arm is shorter because it only consists of two genes. Now if the inversion is occurring here, how will it occur? It will occur in a way that this first arm now becomes longer because now it has to have the three genes and the second arm becomes shorter. So here I have A, then here I have D, here I have C, and here I have B, all the inverted ones, and then I have E. So this particular inversion has taken place here, and this involved the centromere, so we are terming it as pericentric inversion. Now there is a G bending pattern. G bending pattern are those from where we use the Giemsa stain just to identify the bending patterns in the chromosomes, and the G bending patterns have revealed that the human chromosomes differentiate from the chimpanzee chromosomes because of these pericentric inversions which involve the centromeres also in them. Okay, now we should move on further. We have to talk about the inversions in meiosis also here, and these diagrams we have to discuss. Before that, let us see the effects of inversions. There is a effect which is referred to as position effect. Now what happens, let us read this text. Individual organisms with inversions have neither lost nor gained any genetic material. Yes, because only the inversion has taken place, just this DNA sequence has been altered. Nevertheless, these mutations often have pronounced phenotypic effects. There are phenotypic effects, so this is an important line. An inversion may break a gene into two parts, with one part moving to a new location and the destroying the function of that gene. See, there is no part deleted, but the chromosome can be divided in the progenies, and we will discuss it how it can be done. Even when the chromosome breaks are between the genes, phenotypic effects may arise from the inverted gene order in an inversion. Many genes are regulated in the position dependent manner. Suppose if A, B and C are located together, so this B will function only if A and it is located in between A and C. Now if this order gets reversed or if something gets inverted, that time the B gene will not be in the close proximity to A and C, means this order will not be maintained, and in that condition this B gene won't be able to express itself. Now this effect is known as the position effect. Now here also we are concerned with the meiosis only. We have to see that how the two chromosomes which have the inversions, how will they align in prophase 1, how will they do the crossing over, and how will they form the gametes. That is what we are concerned here of. Now see, as I was talking about this, that the G bending has revealed something. So here we talk about the chromosome 4 of humans and the chromosome 4 of chimpanzees. We can see that there is a kind of pericentric inversion here which involves this centromere also. Okay, now we should move to the first part, that is the paracentric inversion. Let's try to not complicate this. Let's try to understand it in simple terms. What is the paracentric inversion? Now, paracentric inversion is that one where the centromere was not involved. Okay, let us take the two chromosomes here, and this being the heterozygous one. So, in this heterozygous condition, there is chromosome 1 and another copy of the same chromosome 1. But the genes have been altered here. CDE, but here it has become EDNC, and that also 180-degree inversion. This is your paracentric inversion. Why? Because the centromeres were not involved. Now what happens is these two have to align together, and what they need to do after this, they need to do the crossing over. So how will they do the crossing over? In the prophase 1 they have to align themselves. Now see, this has CDE in the reverse sequence. So this chromosome, if I name it, let's name it as I have to just decrease the length of my color. Yes. uh, let's name this as 1A and let's name it as 1B. Now 1B has the inversion here. Now they have to arrange themselves into 1A and 1B. Suppose this is the part where the crossing over has take place. Crossing over has to take place. So in the terms of inversion here, the inversion loop will be formed. Now the chromosome 1B has the inversions. So it has to align itself in such a way such that the inversions can be matched with the chromosome 1, or the genes can be matched with the chromosomes 1. So because it has the inversions and also that it has to match the gene order with the chromosome 1, it has to invert itself and form a anti-loop so that the crossing over can take place. So rather than going like this, it is going like this. That is what I wanted to explain. Once the inversion loop formation takes place, then the crossing over has to take place. And if we zoom it into here, yes. I think it's lagging a bit. Uh, okay. Uh, Let me zoom it again. So yes, as I was saying that the inversion loop has already been formed and now it has to do the crossing over. For doing the crossing over, it has to have certain things. That is, the gene order must be correctly arranged, as like if one is directly arranged below the other. So that has been done. Now the crossing over will take place. Suppose this particular arm of the chromosome and then this particular arm of the chromosome, this arm, they are making this crossing over. So now what will happen is the inversion is there and the crossing over has taken place, so it will result into structure like this. Now see, this structure is very peculiar to paracentric inversion only, not the pericentric one. So please remember this. In the paracentric, what happens is, now here is the resultant structure. One of the chromosome consists of two centromeres, and this kind of chromosome is known as the dicentric chromosome. And the another chromosome, see this segment, it does not have any centromere, and this is known as acentric chromosome. So one of them being dicentric and one of them being acentric. Now after that the crossing over has to take place, that has been taken place, and then this kind of structure has to form, this has also formed. Now in the anaphase 1 stage, the two of them have to separate from each other. While they are separating from each other, this chromosome will be lost. Why? Because this does not have any centromere, and because there is no centromere here, the spindle fibers would not be able to attract it. In that condition, this would be eventually lost in the cytoplasm and would be dissolved, and then we will only be left with this particular chromosome which is dicentric, and this bridge is your dicentric bridge. Now after that we have to form the haploid gametes. So after meiosis 2, the haploid gametes will form, and in this what will happen, you see, one of them being the normal one, suppose this is 1, the fourth gamete is complete inversion, so it will not be functional, and the second and the third gametes have certain deletions, so again these will also be not functional. So only one of the gametes remain functional, but the other three gametes become non-functional. So this is your simple case of paracentric inversion, crossing over, and then the gamete formation. Coming to the next one, where we have the pericentric ones. So in the pericentric one, as I told you, not to complicate it further, we have the centromeres involved. Now if the centromeres are involved, so suppose this inversion has taken place here, C, D and E has been reversed in this way, and here the centromere was also involved in between. Now again the inversion loop will be formed, and this time the centromere is also present in the inversion loop, and the chromosome which is below, or the inversion chromosome, has to align itself in a way that the genes can be matched with the chromosome 1. So it has formed an anti-loop. Now again the crossing over will take place, and this time this kind of structure will be formed. Now if you try to observe it very carefully, this is a normal chromosome. See, both of the chromosomes are very normal. None of them is dicentric and none of them is acentric. Both of them are very similar chromosomes and very normal chromosomes. But if you see, after anaphase 1 and anaphase 2, what happens? The two of the gametes become normal, and the two of the gametes are non-recombinant gametes, but the recombinant gametes have the inversion sequences, and because of that they become non-viable, and they will not be able to produce anything like that. Okay, so this is what the case of paracentric and pericentric involves. Okay, there is one more thing here. If you see this gamete number 2, see, gamete 1 is normal, there is no doubt in this. Gamete 2, what happens here? In gamete 2, there is a kind of recombination, because of which it has become non-viable. Why? Because one gene is duplicated here, and the required gene now here is not present in this gamete. So this will become non-viable. The remaining two gametes, if you see, they are also non-viable. Why? Because they have the inversions. So obviously due to inversions and due to the position effects, they will become non-viable again. So this is the condition of paracentric and pericentric. In the paracentric also, one of the gametes was only viable, which was non-recombinant. The other three became non-viable because they had the deletions and they got some of the genes got missing. But in this one, all the chromosomes look normal from the length wise, but they had the inversions, they had the certain genes which were duplicated. So that's why they became non-viable, and only one of the non-recombinant gamete was viable. So this is what and how the inversions can take place. Now lastly, we have to discuss about the translocations in the structural chromosomal structural arrangements. So translocations are basically the rearrangements in which the segments are exchanged between non-homologous chromosomes. Now we should not move into much details of this, but yes, you should know that these translocations occur in a way that, let's say, assume this is one chromosome and this consists of A and B and C gene, and this chromosome consists of P, Q and R gene. So what happens, this B and Q, here the inversion will no sorry not inversion, translocation will take place, and the resultant chromosomes will have A, Q and C, and here they will have P, B and R. So this is a simple kind of translocation. Now the translocations can be of two types, one of them being the reciprocal translocation and one of them being the Robertsonian translocation. So what happens in the reciprocal translocation? There is a mutual exchange between the two chromosomes, and it is the most common type. At the anaphase, the segregation may be either adjacent also, or that may be alternate also. Now see, let me show you the diagram here. I think the diagram is not provided. Nevertheless, so basically what I mean to say in the reciprocal translocation, this whatever I have shown here, this is only the reciprocal translocation. It is quite normal. Now this reciprocal translocation wherein there are two chromosomes involved and there is a mutual exchange of genes between the two chromosomes. Now when I talk about the Robertsonian translocations occur in the acrocentric chromosomes. Acrocentric chromosomes are those chromosomes wherein one of the arms is very short and the centromere is towards the end of one of the arms. In the acrocentric chromosome, when they will do the exchange and when they will do the crossing over, what happens is that one of the chromosome becomes extremely long and it becomes metacentric in nature. The another one of them left in fragments. This fragment is eventually dissolved and is lost in the cytoplasm. So here, due to the translocation, one of the chromosomes got lost after crossing over. So this is your Robertsonian crossing over, so or Robertsonian translocation. uh, let us try to understand this diagram here. So this is an individual heterozygous. For this translocation possess one normal copy of each chromosome. See, there is one normal copy of chromosome, let's say N1 and N2. N1 and N2 are the normal copies and T1 and T2 are the translocated copies. Now because each chromosome has sections that are homologous to the two other chromosomes, a cross-like for configuration forms in prophase 1 of meiosis like this way. So here we can see that there are, this was N1, this was N2 and this was T1 and T2. So they have formed this kind of structure. This is a cross-like structure. And after that, during the anaphase, they can do the segregation. Now the segregation can be in the alternate way or it can be in the adjacent way. Always remember, when the segregation is in the alternate way, you don't need to have much details of the diagrams, but do remember that if the segregation is in the alternate way, the gametes will be produced viable, and if the segregation is in the adjacent way, the non-viable gametes will be produced. So this is a typical example of how the translocation effect is visible in the non-viable gametes after the anaphase 1 and anaphase 2. Now there are certain examples related to translocation. One of them being the Down syndrome. I had this example here only. Okay, so in the Down syndrome, what happens? As I was telling you, the Down syndrome involves the Robertsonian translocation. Now there are two normal copies of chromosome 1 present and one extra long arm of chromosome 21 is attached to another chromosome. So always remember this, in the trisomy of 21 chromosome in the Down syndrome, what happens are there are two chromosomes 21 which are normal, but one chromosome which is like extra large, and this is all because of the Robertsonian translocation which can occur here. After that, we should talk about the fragile sites. So these are those regions of the chromosomes that appear as gaps or breaks under specific conditions. What is the meaning of fragile site? This is a chromosome. Suppose this part of the chromosome is a fragile site. Now If we talk about this fragile site, if we try to observe it under microscope also, we will see that this site consists of certain gaps in between like this, and then the normal chromosome. So this is your fragile site, which can be observed in the chromosomes. Now there is one very specific syndrome here, which is Fragile X syndrome. So Fragile X syndrome is observed in certain females as well as even maybe in the males. So here what happens is, with CGG, this repeat occurs in the FMR1 gene, FMR1 gene on the X chromosome. So this can also occur here, and this is also one of the examples which is due to translocations. Now we have to talk about the aneuploidy and polyploidy. Let's take in the another video that topic. In this video, we should only talk about the structural rearrangements. So this is all from my side for structural rearrangements. In the next video, we should talk about the wherein the chromosome numbers get altered.